First of all, I'd like to thank the organizers for this opportunity. Today, as Gina introduced, I will talk about nanopesticides from the development until the mechanism of actions in crop protection. I'm from the Institute of Science and Technology of Sorocaba from Sao Paulo State University. Regarding the agriculture, it's important to consider that agriculture is a human activity, and as a human activity, it's considered economic characteristics, and the idea is that you can develop agriculture to promote or to produce food to the society. However, it's important to consider the environment. Sometimes the actual models in agriculture do not consider the environment as a key issue. So to promote sustainable agriculture, we needed to think in these three pillars together. And how to consider these three pillars if the actual scenario, in special the model that we are using in agriculture nowadays, it's from the 1950s with the Green Revolutions. During the Green Revolutions, we had some actions. We used mechanization, chemical farms, Green Revolution, and plant breeding, and the biotechnology. And what was the results of these actions? We have now pest resistance, we have food contaminations, we have loss of soil fertility. So we have a lot of problems regarding the environment. And this is not a model that we needed to use anymore, as we needed to promote a more sustainability in the agriculture actions. And to produce a more sustainable agriculture needed to decrease the amount of synthetic pesticides. We can use, for example, plastic culture. We can use the biological control and the plant-based pesticides. What we expected with this kind of actions, we expected that we can restore the soil biodiversity, that we can increase the crop yields, decrease the pest resistance, and increase the food security. Another important point is that actually agriculture is responsible for the 70% of uses of the groundwater. It's responsible for 30% of energy consumption, 75% of deforestation, 30% of greenhouse emissions, and 30% of land uses. Also, it's important to think that now we are preparing the world for the next years, and it's expected that to feed 10 billion people in 2050, we needed to increase the food production around 60%. And it's important to consider that some countries will be responsible for this increasing in the food production. And I'd like to show this data from the USDA report, where you can see that Brazil, it's a key country in the world to produce more food in the future. The question is that I'm not telling here about the increased land to produce more food. We need to preserve our forests, we need to preserve our biodiversity. What I'm telling here, it's we need to increase the amount of food per land. And to do this, we need to have integrated and new technologies. In this context, nanotechnology in agriculture it's a kind of focus nowadays, because we can see in very nice journals such as Nature Nanotechnology or Nature Food. In the last 2 years, they are publishing a lot of papers about this topic. And especially, I'd like to emphasize this paper from Nature Foods, where the authors described what is the stage of the technology regarding the nanotechnology in plant produce. It's important to consider that the nanotechnology can be used as nanopesticides. We can use nanosensors to check the stage of the plant developments, or if you have some pathogens infecting some plants. It's possible to use nanotechnology as nanofertilizers. It's possible to apply the nanotechnology in genetic engineering. We can use it in seed coating. So it's a lot of possibilities to use nanotechnology in agriculture. Regarding the biopesticides, that is the focus of my presentation, why we can use the biopesticides? Usually they are less toxic than the conventional pesticides. Generally, they affect only target species or closely related organisms. It's different from the synthetic pesticides that they affect a broad range of organisms. And most of times these biopesticides, they are effective in very small quantities. And this is interesting because they can be decomposed quickly in the environment, and it's not persistent. So the idea is that we have a lot of natural sources of molecules, and using these molecules as bioactive molecules, we can improve the pest control and promote a more sustainable agriculture. What my group is doing in this direction, we have a project, an ongoing project that the name is Nanotechnology, Agriculture, Nanotechnology and Environment. This project is sponsored by Sao Paulo Research Foundation. And the idea is in this project that it can encapsulate the essential oils or essential molecules, that its components from the essential oils with nanoparticles to promote some pest control. And also we can have another kind of biopesticides that use organisms like bacteria or fungi, and we encapsulate these in microparticles to promote some biological control in agriculture. And today I will talk about the nanopesticides. This topic we reviewed from the last 5 years. We are reviewing the topic, and the idea is that you can connect different active ingredients from different kinds of plants with different strategies to encapsulate it and to move to a more sustainable agriculture. The first case that I'd like to show, it's the case of botanical repellents. We select different botanical repellents that it's found in isolated essential oils from plants, and we start mixing these components in a combination that there is no essential oils in nature. So this means that it can combine geraniol and the eugenol, and it's not, there is no essential oils in nature that have the mixture between geraniol and eugenol. And we use different strategies to encapsulate and to use in pest control. The first example, we prepared some Zen nanoparticles with botanical repellents. Zen, it's a corn protein, and it is interesting systems to prepare nanoparticles because we can dissolve them in an ethanolic solution, and we can add here the active ingredients, and we just mixture this under magnetic stirring in aqueous solution with a surfactant. After this stage, we just evaporate ethanol, and you have this suspension with the nanoparticles. So it's a very green way to produce these pesticides, nano biopesticides. Here you can see the size distribution of the particles over time. So we measure the stability of the systems over 120 days. We can see that the shape of the particles are spherical using atomic force microscopy, and we run some experiments in greenhouse conditions. And in this case, we use a red mite as a model organism. It's important to consider that to compare the data from our nanopesticides with the essential oils or the combination of the essential oils, we need to prepare emulsion, as this oil is not soluble in water. So when we prepare emulsion, we can see that the repellents of the red mites, they have a peak and a decrease over time. And when we consider our nanopesticides, our mixture of geraniol and eugenol encapsulated in Zen nanoparticles, we can see that in the small time, there is no big repellents, but over time, we can have some good repellents. This means that in this case, we can't use only our systems or nanoparticles, Zen nanoparticles, or you can't use only emulsions. If you combine both, we can have some of these areas, and you can see a better control over time. So this is important because now we can mixture we can combine these formulations, the emulsion and the nanoparticles with Zen, and we can tune the repellence of red mites. Also, regarding these systems with Zen nanoparticles, we decided to develop some devices using some hydrogels. So we prepared the particles with the botanical repellents in the same steps as I showed before. And after that, we prepared a hydrogel with a mixture with some polymeric materials. We have a paste and we molded these pastes. And after this, we did some reticulation with citric acid. This is the modes that we use, and this is the hydrogel images, and this is some schematic I came from the hydrogels. We have the particles, we have the polymeric materials, we have the acid, the citric acid as close linker. And to check, the idea is that you can develop some devices and you can put these devices in your home, or if you are producing some small food or a small garden in your home, we can check if these systems works. We use a model with 2 vases and using red mites again. And we put this device in a stick and we add this in the vase. So here you can see some images of these experiments. In this point, we add the red mite, and the red mite has the option to come in these directions with the hydrogels or to move to another direction. When you check these biological activity studies, what you can see, the control, the distribution of the organism is the same. The control of hydrogel with no botanical repellents the same, there is no statistical difference. A system with nanoparticle, there is no differences. But when we use the emulsion with, for example, geraniol and cinnamaldehyde, or nanoparticles of zinc with geraniol or cinnamaldehyde, what we observe that the amount of organisms in the side from we to have these sticks with the hydrogel, it's very low compared to the other side. Also, the same happens with emulsion and nanoparticles incorporated in the hydrogels with a mixture of geraniol and eugenol. So we can see the data here, and you can observe the data in the untreated plants. With these results, we can calculate the repellence index, and you can see that the repellence index is very high. So this is very nice, and this is interesting systems to be used in crop protection. Sure that it's not used in huge fields, but in small farms and at home, it is possible to control some pests. Another advantage of this hydrogel, the system is not sprayed in the plants. So this means that the repellent is not in contact with the food. Another kind of trend is that we have in applications of agriculture, in nanotechnology in agriculture, it's the idea to use some stimuli responsive factors to release the active ingredients into the environment. We can consider the abiotic and biotic stressors. For example, we can use the temperature, fluid, draw, salinity, or heavy metals that can connect it to the nanoparticles and promote some release. Also, we can consider the presence of fungi, insects, weeds, or nematodes. To follow this idea or these trends, what you did, we create a hypothesis that we can prepare some protein nanoparticles like I showed before with Zen, and we can associate in these Zen nanoparticles botanicals, insecticides, and you can spray these in plants. And when we spray these in plants, the organism can eat the leaves of these plants, and during the digestive process inside the organism, proteolytic enzymes can degrade and release inside the organism the insecticides. So the idea is that you can direct the organism to the target organism. So here you can see some characterization of these nanoparticles. We prepared the design nanoparticles with some bioinsecticides. We characterized by nanoparticle tracking analysis, by DLS, by AF atomic force microscopy. We can see that the particles has a medium size around 100 nanometers. Also, just in order to check if the formulations can be toxic or not to plants, we run the phytotoxicity studies. And over different concentrations of the systems, there is no effects in the shoot and the root. This means that these formulations can't promote some toxicity to plants. Just to check the proof of concept, we run it at the first stage in vitro studies. We have here the formulations, and we incubated the formulations with enzymatic, proteolytic enzymatic materials. And over time, you can observe that there is a decrease in the Tyndall effect. And when you calculate or measure the size distributions and the concentration of the nanoparticles, you can see that the concentration of nanoparticles decrease over time. And the explanation to this decrease in the concentration, it's because the particles are being degraded by the proteolytic enzymes. Also, we run a protein gel electrophoresis, and here you can see the protein, that is zinc, that we use to prepare the particles. This is the protein bands. This is the nanoparticles, so we have the same bands of proteins, and we incubated the nanoparticles with trypsin and over time. And what you can observe that is the bands from the protein, it's decreasing, and it starts to appear some small fragments of proteins, and these small fragments, it's probably due the degradation of the broken or the breaking of the nanoparticles due to the activity or enzymatic activity. Also, we decided to check the mechanism of action in vitro proof of concept, and what we did, we needed to track the particles inside the organism, and we labeled the zein with a fluorescein, and we did some cross-linking between fluorescein and the zein nanoparticles. And we offered these particles in a diet to insects, and over the time, we consider the degradation of the particles because of the proteolytic activity, and we quantify the fluorescence probe inside the midgut and in feces. So here you can see the amount of fluorescein over time inside the midgut, and you can observe that there is an increase over time, and also there is an increase of fluorescent probe inside the feces. Also, we observed the midgut images. Here is the bright field of midgut, and this is the fluorescence field. There is no background or fluorescence. And over 2 hours, we start observing, after we feed the organism, after 2 hours, you can observe a strong concentration of fluorescein probe. And over time, you can observe that all the midgut, it's labeled with the green probe. So this means that the particles are here, it's starting the digestive process, and over time the proteins start degrading by the proteolytic enzymes inside the organisms. Also, to check if the enzyme can promote the release of the fluorescein, we have a situation where we incubated the nanoparticles with the midgut and the trypsin. We calculated, or we determined the activity of this the proteolytic enzymes in these solutions, and we can see that when we encapsulate, or we put together, the nanoparticles with the enzymatic content, to have the same profile of release of the fluorescein. And when we compare with the absence of enzymes, there is no release of the fluorescein. So this means that the proteolytic enzymes inside the midgut is responsible for this degradation of the particles. So, as conclusions, I'd like to emphasize that nanotechnology has potential to be used in agriculture, especially in sustainable agriculture. And it's very important to consider plant-based insecticides and repellents in combination with nanotechnology to crop protection, in special with focus in target organisms. However, it's important to consider that we are starting this technology or the use of this technology in agriculture, and we need to have more information about toxicity, the fate of these pesticides, the mechanism of actions, and to check the food security. To finish, I'd like to thank my co-workers and some postdocs and master and PhD students. I would like to thank the sponsor agencies, Brazilian sponsor agencies, CNPq, CAPES, and FAPESP. And I would like to thank all of you for your attention.